Well, welcome everybody. It's a real pleasure to be here, and thank God I believe in modern medicine because I've been having body aches and shivers and all of this today. So I know people expect me to be funny and entertaining, but I'll have to see how far that goes tonight. But I'm really happy to be here. So the work with CDI actually started at least 3 years back from where we're now. And it was actually quite an interesting story, and I want to share the story with you because some of you might not know about it, some of you may actually can resonate with this. So I was hired into Pfizer about 8 years ago, and our main attrition was really hepatotoxicity. So we had lots and lots of people, actually hundreds of people, working on drug-induced liver injury and trying to build assays, right? And we actually managed to build an assay, or we thought we built an assay that could predict human hepatotoxicity. And back then I was in La Jolla, and La Jolla converted into oncology, and our product suite and other kinase inhibitors. And before we knew it, the hottest thing on the horizon was cardiotoxicity. And people said, well, you got to do something about this, you really got to fix this. And I'm like, okay. So back then, about 3 years ago, we really thought that if we just had a cell line that was very close to representing the organ of toxicity, so in this case the heart, we predict cardiotoxicity. So you think that's true? Alright, I'm not going to put anybody on the spot. So yes and no, because what we were measuring back then was mechanistic endpoints of injury, such as mitochondrial toxicity, and some of you know me as being that person, oxidative stress, ER stress. All of these endpoints are mechanisms of toxicity, but they're not exclusive to liver or to cardiac toxicity, okay? So this was a big learning we had to do, right? And so when we started out with CDI, we really thought, wow, there's a human cardiomyocyte, beats, and we can use it to predict cardiotoxicity. And so what we had proposed was really, to go ahead and do some high-content screening and look at mitochondrial toxicity and ROS formation and membrane potential. We thought that was all going to fix it. We realized that that was not the case, right? Because if you take doxorubicin and you put it in a hepatocyte, it will flame up, and vice versa. So it wasn't as simple. So we got together, and this has been the nice thing about our relationship with CDI. Blake was the first one who says, okay, I get it, Yvonne, I get it. What are we going to do now? So we got together and we really thought, so what is it we can do? And one of the things that I was always interested in was to even understand, what is a cell? What does it do? And what does it need to be doing for me to see what I want to see? And so, for example, in order to see mitochondrial toxicity, you need to have mitochondrial function, right? Well, you're not going to have mitochondrial function if you culture them high glucose. They can upregulate glycolysis, and you give them a mitochondrial toxicant, and they say, nah. Doesn't bother them, okay? So one of the things we went back to, talking to Blake was, maybe we just need to start real simple. Maybe we need to just understand what we have. And once we understand what we have, then we can figure out what we're going to do with it, okay? So we took a big step back, and what we ended up doing was we wanted to take a look at the bioenergetics. So we wanted to understand what makes them a heart cell. What is the substrate they actually use? So anybody knows here what the adult heart uses? That's right. Do we culture in fatty acids? No! I don't even know what that is, right? So perfect. So that was one of the studies, and there's Payal sitting right there. And I'm very proud of Payal's work, who has done actually an extensive bioenergetic study, and I'm going to show you a couple of her results. And then the other thing we wanted to know was, so if we culture them, how close are they to the heart? Do they have what the heart has, which are ion channels? Are there still stem cells? We don't want stem cells, we want adult cells. Are there still stem cell-like? And so we have Dinesh right here, who spent a lot of time really characterizing, by gene expression, how these cells look like. And I think what we came to is a really good basis of understanding what the cell is, who the cell is, and so our next step going forward is to figure out what are the applications. You've seen many walking around the posters. I think there's a great potential for some of the beading applications. These things bead, right, Blake? And so we know that. We now know not to culture them in glucose, right? And I'm taking the punchline away from what I'm going to show you. We know that we can utilize them to do some transcriptional analysis to figure out what is actually going on when I have a compound that's toxic in human and I can't see a thing in these cells, right? and vice versa. So lots of utility, but we've actually really taken that step back of saying, can we characterize, can we understand, can we get comfortable with our new friends, the iPS cells? Okay? All right, so I'm just going to show you a couple slides of what we have done. And again, I deserve no credit besides getting it all started and causing all the trouble. But it's really, Payal and Dinesh are in the audience. So if you have any questions later, don't come to me. Just go right to them. You have done a fabulous job. So, okay? All right, so back to the story I already told you. So I make slides, but I rarely ever talk to them. So this is just a scenario of telling you that, besides liver toxicity, cardiotoxicity has really become a major point for attrition, black box warning, limitation of dosing in the clinic. So this is not something simple, and even the FDA demands that some kind of cardiotoxicity testing needs to be done. Okay. So this is actually one of my favorite slides because that's the first thing I got hammered into me when I started at Pfizer 8 years ago. You know what we did? We did exclusively look at how a compound behaved in animals, and we already know that translational piece into humans doesn't always work, right? But we utilized happily, rat studies for SCR approaches of compounds. And what we realized was that if we failed really late in the game, this just wasn't good. And this is just to show you what this all cost. And so we really tried to shift the entire paradigm, and that actually caused my move from beautiful La Jolla into rotten Grotten, as I call it, was trying to really drive. Oh, please don't record that. Too late. I know. I figured that. To really try to drive safety much earlier into the organization, because what we actually knew, for every dollar you invested, you saved a lot of money later on. And so this is the way we think about it when we think about drug discovery. We have the target selection, series selection, lead selection, and candidates, okay? And this is a recent expert opinion that Craig Thomas and from Eli Lilly and myself wrote. And what you see is the way we think about it. We think about targets, and you can look at it and you can say, wow, does this target potentially have liabilities? And that's what you know from knockout studies and antisense and just a really good, PubMed or competitor search. You want to know, is the tissue and species specific, right? So if you put in the rat and it's not in the rat, then it's not going to help you. So series selection, that's where I'm actually now working most of all, is, we're putting some in vitro high cell-based screens in for general cytotox, gene tox, but also thinking about organ tox. And I think I mentioned, that this is not trivial to really detect organ toxicity. I'm not saying there is no such thing, because we know HERG relates to cardiac and not to liver, and BSEP relates to liver, not to heart. But there's many mechanisms that are in common. So predicting organ toxicity is not trivial. But what I've highlighted here in red is, the points during this drug development process where you can think about using, the cardiomyocytes and the cardiac evaluation to get a better understanding. So, from simple screens to looking at ion channel pharmacology, to later on doing the in vivo safety pharmacology. So we've made a lot of progress with some of this, but we need to pull it together so that at each and every stage of the drug discovery process we can have something in place. And some of it needs to be applicable to thousands of compounds, versus others, has to be deep biology on a particular compound. So, but there's many ways we can think about it. But once we think about it, we need to know what we actually have in our hand. Okay, so as I said earlier, I've given a lot of thought about what we actually want from an in vitro model, and that's an unlimited source of cells. We want them fully functional, so for liver cells, that would be phase 1, phase 2, BSEP, and so on and so forth. And in the heart, we want it to beat, we want it to have channels. And I think, maybe I'm wrong, but I think we know a little less about the heart than we know about the hepatocytes. So I think there's still a lot of learning. The assay ideally should be reflective of a discrete organ toxicity, and I told you about the caveats there. There should be a clear connection between the in vitro data outcome and the plasma concentration where the toxicity first was observed. So very often, you screen and you get IC50s, and you don't know what the Cmax is. Could be very different. So the rank order could easily change, right? Same from a potency point of view. Assay should be tuned to sensitivity. In addition, organ toxicity is rarely characterized by a single event, right? It's a whole cascade of things that can contribute to it. And some of the mechanisms, as I said, are not very specific for particular organ toxicity, such as mitochondrial dysfunction, oxidative stress, and ER, and that was a hard lesson I had to learn, but I've learned to live with it. So it's all good, so you can leverage it actually across a lot of other things, so which is actually good. So these are the projects we've done at Pfizer. We have done the characterization of the iCells over a long period of time. And we've done that with respect to the metabolism, the bioenergetics, and the expression of adult cardiomyocyte-specific genes. And once we've done that, we now are thinking about, how can we test the utility to detect cardiotoxicity in many different ways. One is ion channel, one is not ion channel mediated effects, and then effects on beating. So 3 different ways we can think about it. And then build in vitro and vivo correlates and further investigate the false negative space with gene expression studies. And what does that mean? So this is really one of the things that Dinesh is interested in. There's many compounds. Everybody who's ever done cell-based assays knows that our predictivity ends about 50%, right? So there's a large false negative space. These compounds look beautiful, and you get in the animal, and the animal falls over. So there's a lot of potential to use, more sensitive assays, gene expression, to say, wow, can I get a hint of what's going on? All right, so this is the bioenergetic characterization we did. Payal has a poster tomorrow. She just wrote up the paper. She's done a boatload of stuff. But really all I'm going to tell you today is about, what she found out when she called it in the different media. So we're talking high glucose, low glucose. Low glucose was fatty acids, galactose was fatty acids. And we'll show you some gene expression that she did over that time and some testing of kinase inhibitors. Okay? So there's more work that she has done, and you can come and see her tomorrow or talk to her later. But I'll just have a couple of examples here. So I think what we were trying to talk about here is the fact that if you culture in glucose, cells are glycolytic, there's not a lot of oxphos activity, they're not very aerobic. That's not the heart is aerobic, right? And we talked about the fact that the adult heart uses fatty acids as a major fuel. And so what you see here is simply on day 7, day 14, and day 21, the oxygen consumption over the glycolytic rate ratio in the high glucose, low glucose, fatty acid, galactose and fatty acids. And I think what everybody in the audience can see when you come to the galactose and the galactose fatty acid, that rate is much, much higher. So they generate really the ATP through the mitochondrial function rather than the glycolysis. Interesting, what you also see is that we do get an increase over the 14 to the 21 day. So there's some kind of additional maturation happening. And it looks to us that 14 to 21 days, at least from our point of view, if you need bioenergetics, then that is really the time point you want to do your experiments. Okay? Can everybody see that? All right, perfect. Okay. So this is the next piece where Payal really looked at gene expression using customized PCR, and really, again, just very focused. She's just looking around oxphos activities. She's looking around glycolysis. And then one of the things that she and I had talked about was, one of the things that I had heard through the grapevine was that cells do change in culture all the time, and that they have to actually upregulate some of the antioxidant activities in the apoptotic machinery in order to survive. So when she did the comparison to the heart, that's exactly what she saw. She saw that in general, even though, as I just showed you, we had quite a bit of bioenergetic function, but in comparison to the heart, to the human heart, there's definitely much, much less activity, and that doesn't, really surprise us that much for cells in culture. But then again, also confirmatory, we found that some of the glycolytic and oxidative stress and apoptotic genes were actually upregulated. And so this is gene expression. I was trained as a biochemist, so I would say, well, you're wrong, I don't believe you, and that's just a gene, and it's PCR, and full difference, and whatever else I can come up with to make them miserable. So Payal, of course, went ahead and did the protein expression. And what you see here is, again, starting from the left, going to the right is high glucose, low glucose, ending up with the fatty acids. And then the heart in the very right hand of panel A there, you see that in comparison to the heart, there's definitely much, much less protein of the oxphos, of the mitochondrial chain. But when you look at the galactose and the galactose fatty acid, you see that there is actually an increase in that protein. Okay? And then when you go over to panel B, looking at SOD, BCL2, you actually see that in the heart, they're not there, and they don't really need to be there unless you have a pathology condition, but this is healthy adult human heart. But what you see is that in these cells, some of these are upregulated. And that is something, what people have commonly seen. And so, there's been people talking about, adding NAC and glutathione and ascorbate and other things to media, and it's something one can think about it. But okay, so this was another fun thing we did. That's how it all started. That's how my phone rang. Do something about Sutin, do something about sorafenib, they all have cardiotoxicity. And so here, what Payal did is just took a look at all of these kinase inhibitors, and what she found was that some of them significantly decrease oxygen consumption, such as sorafenib, and then others do it much, much less. And she did this in galactose plus fatty acid, because that's where we found we actually had, the OXPHOS capacity. And so what you don't see here, but what we know is that a cell on sorafenib wouldn't really survive under these conditions because there's no more ATP production. What you can do now is, this is the rank order as it exists here in the cell-based assay. And so now, but what you have to really look is your exposure. Sorafenib could look very potent here, but if there's a very, very low Cmax, then that TI could change. The other thing too is these are oncology drugs. So for sorafenib, for example, this might actually be efficacious, right, to attack some of the mitochondrial functions. So a lot more work has to be done, but this was just something we wanted to, to try out to see if we could actually generate those response curves here, and we could. Okay, so the conclusion here is really what I already told you, is that, iPS cells really look a lot more aerobic when we put them in galactose and fatty acids. We do know that they somewhat mature, if that's the right word to use, but they stabilize around 14 days. What I will show later for Dinesh, and we just talked to other people, you can culture the cells for a very long time, but this was done on the COS platform. It's a very specialized plating, so our limit really was more like 21, 28 days, but we do know that's a specialized platform, and we do believe that you probably can push this out quite a bit. We do know that these cells in galactose and fatty acids are much more susceptible to mitochondrial toxicants, and so we know that they rely on OXPHOS for ATP. And then the gene expression confirmed that these cells are capable of adjusting to different carbon sources. Actually, we should go back and I should show you that there was really not a whole bunch of difference between the different media here. So, and back here. All right, what else? And our screen of kinase inhibitors showed that these cells are, a suitable cell-based model that can be utilized for toxicity screening. And again, it was everything I already told you of how you might have to actually look at this in the big picture. All right, so this is Dinesh's work, who looked, as I told you, on the gene expression of the iCell cardiomyocytes up to day 42. So you had a lot of work to do. So RNA samples were taken at all these time points, and chips were run, and gene expression analysis was performed. And then, as I said, Dinesh, of course, compared this to cardiomyocytes. So this shows the gene expression from day 2 to day 6, and it kind of confirms what we had seen before too. Blue would indicate significant changes, and red insignificant changes. And really, what you see here, there isn't really a change, at all from day 2 to 6. However, when you go from day 2 to day 14, and that's kind of where we saw a big jump too, there's a lot of genes that have significantly changed at that time point. So very nice piece of just looking at the significance values here. And this is a heat map that Dinesh has generated, and what you see is here from the top right to the bottom, you see all the days of the iCell cardiomyocytes, and then you see how he references the adult heart and the fetal heart, because that's very important too. We want an adult phenotype and not a fetal and then also some skeletal muscle. And that's interesting because very often people are worried about that some of these heart cells actually would also have a skeletal muscle type phenotype. And so here's basically a couple different markers. So to the left here's a whole bunch of stem cell markers, and of course you want them to go away, right? So they're red. Red means low or no expression. And then you have all these cardio markers, and you would like them to see, go up and be stable, right? Go up first and then be stable. And you can see that they're all yellow and green, so that's medium and high expression. And when you actually look from day 14 to day 42, you can see that there's actually no difference versus if you, again, like I said in the previous two slides, if you go from day 2 to day 6, you see that a couple of the yellows are becoming green, you see that? But then later on, there's really very nice consistency, and you can see that in comparison, that it's very comparable actually to the heart. And then there's some skeletal muscle markers, and again, we don't really want them, and they're not there, so that's fabulous. And same with some neuronal markers. So a nice piece of work showing, the consistency or stability of this model from at least day 14 to day 42. Very nice. And then you can't see the fine print, neither can I. But, we all know what Dinesh was after, right? Dinesh was probably not after, a glycolysis gene. What was Dinesh after? Probably all kinds of channels, right? And all kinds of channels that should be in the heart, and all kinds of channels he couldn't detect that shouldn't be in the heart, but in other tissues. So really a lot of cardiac markers, and again, Dinesh is here, so you can have a little chat with him. So really nice piece of work.
I just wanted to ask what that stem cell marker was on. There's one stem cell marker on the left there that seems to be persistent.
Dinesh, I'll have you answer that yellow little guy down there. That's in the skeletal muscle though, not in the bottom is the skeletal muscle.
No, it's the one on the yeah. All right, you guys can look at this later. So, all right. So the conclusion here is no significant changes were observed between day 2 and 6, but definitely between day 2 and 14. And the expression profile was very stable. So even I was surprised because I'm real skeptical when it comes to that. They lost their stem cell markers, that's good, and displayed medium to high expression of the cardio markers. As I said, Dinesh looked at very specific things such as HERG, NaV1.5, structural markers. So it's just a really solid characterization. We're not saying there isn't something that's missing or low or high or different from the heart, but this is just really a start of getting a feeling for it so we can optimize for it. And not just optimize, but, do a fit for purpose. So another thing is if I have a NaV1.7 program and there's no NaV1.7, then there's probably no point of using the cell. So we're thinking more, the more thorough you really characterize the cell, the less surprises you have, right? And they do lack the expression of skeletal muscle and embryonic and stem cell and pluripotency markers. So I think that's it. Yeah. Any questions?
Any questions? If you don't mind.
This is a question for anyone, Yvonne or Blake or anyone known. But one of the real challenges we have in toxicology is, as you well know, is we see a toxicity in a preclinical species. Let's say we have liver tox in a rat. We don't have liver tox in the cell. We want to know if the rat is predicting the human or not. Is it possible to make pluripotent stem cells from other species and then do a cross-species comparison?
Yeah, absolutely, absolutely,
yes,
yeah.
yes,
That's why I asked earlier if he can make me in the dish, right? So yeah, and
well, I presume you're a human. I'm talking about other species.
Don't you dare be presumptuous.
Good one, I like it. I think you have a point there. I think I'm cautious about that. So I can speak for hepatocytes much better than for cardiomyocytes, and maybe you can enlighten me about cardiomyocytes. So when I think about hepatocytes, right, I know that CYPs are different. We know there's surrogate CYPs for human CYPs and rat, but the distribution of these CYPs is different. We also know that there's a lot of structural homology between B-CYP, between rat and human, but when you take a rat hepatocyte, the distribution or the amount of B-CYP present is different from human. So I have a lot of knowledge in that field to be able to slowly characterize it, and then, you know, when you do your comparison, you're not just saying, oh, it's more sensitive in the rat, but you can say it's more sensitive because it's a B-CYP and there's more B-CYP, right? Or it's more sensitive because it's, you know, 3A4 and that is how it is. So in the heart, you know, I'm just starting to learn this about how much difference there is between some of the ion channel pharmacology. If we look at structural type of cardiotoxicants, and again we're talking about mitochondrial oxidative stress, ER, lipid accumulation, lysosomal, I assume that there's less than 5% difference between the species. You agree or you disagree?
Well, we clearly do have species-specific toxicity, regardless of how you measure differences among species.
Right.
We have to explain that somehow or other. I mean, it's a fact.
Yeah.
But what about the? Do you have?
So,
do you have, or are you thinking of making?
Yes.
Cells from other species?
So we have cynomologous ES cells, and we're in the process of generating some additional species like beagle to be able to do that exact study. The problem and the issues that have arisen earlier have been that some of those species, like canine and others, including rat, were very difficult to produce iPS-derived cells from. And so a lot of the work was done in mouse, obviously. But I think you'll see some of that data being generated because we get that question a lot, and whether it becomes a product or just a data set to justify the correlation such that they can be moved further in the productivity process, it remains to be seen.
So again, I don't know much about cardiomyocytes, and I have no problem admitting that. But interestingly too, and I think about these things again with hepatocytes, right? We know that mitochondrial function, you know, is much more affected in human versus monkey versus, and that was studies done in our long time ago. And if you wanted to criticize it, you could only say, well, it took them a while to get the human, you know, in the house versus the rat was right there. So we do know there are sensitivity issues as you go towards primates and human, and I think that's what you're experiencing too. So the interesting thing about this is, you know, we always thought about this, you know, what a rat eats versus what the monkey eats, you know, and again it comes back to that we actually do a pretty standardized culture conditions, right? But I think that the substrate utilization could potentially, at least in the liver, you know, again I can't speak for her, it could be different between a rat, you know, and a human just because of the nutritional status. And until we model that properly, we probably over time lose that species specificity. That's what I'm afraid of, put it that way, right?
Okay, thank you.
So, all right, good to move on?
Yep.
All right.
Another question?
Yeah. Sure.
So when you know, what kind of protocol do you use to do the cycle toxicity for cardiomyocytes? Do you use
Oh, what format?
What format? Well, we personally like to scale up to 384 because we do we sit in chemistry. We try to make the chemist responsible for safety, so they want to know which series is better than any other. So we are looking up to like 2,000 compounds a year for full safety profiling. So for us, it's a necessity to do 384. Traditionally, we have done a lot of 96, even cuvette-based. So if I de-risk a compound and I only want to benchmark against whatever's marketed and it's two others, you can go to any format.
So you find that there are big differences between day 2 and day 14. So when you do the cardiomyocytes, what day you do?
Oh yeah, so we don't do anything till day 14. Yeah, we don't do anything to, we like day 21. We like day 21 very much.
So you will plate
It's my favorite
cells
day.
in the plate first,
Mm-hmm.
then culture them for like 2 weeks, then you do the drug shifting.
Mm-hmm. Mm-hmm. Yeah. 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 Exactly.
And then what would be your assay? What would be your assay at the end of the experiment?
I don't have an assay yet.
So, you're
So,
saying 2,000 comments?
So no, I was speaking broadly for our department when she asked what format we usually go in. So this is what I was trying to tell you up front. I do know that a lot of the parameters we traditionally measure, ATP, glutathione, mitochondrial function, has nothing to do with cardiotoxicity. It's applicable all over. So I can throw triglyceride on these myocytes and they're not going to like it. So I think this model is much more fit for purpose. So what can they do other cells can't do? They can beat. So effects on beating. We just started to characterize the channel activities. There's another potential. If you just want to look at, and they'll probably kill me later, but if you just wanted to look at ATP depletion, you're probably going to have as much luck using any other cell line. And I mean, it's out there in the literature where people looked at 48 different cells and said there's only 5% difference between compounds. So I think we need to understand the fit for purpose of what these cells have. So it's the genetic background, then the question is, can we maintain it in culture, right? So you talked about it, how you could, do any patient. So what's the difference between you and I, right? The channel activity, the beating. So I think we just need to further characterize in the model, and we always like to do this overnight and have all the answers, but I think I've been long enough around to really truly characterize a cell model, the growth conditions, and what I can do. I would say it's a 3 to 5 year project. Would you agree?
Mm-hmm.
Yeah. So. Okay. All right.
Thank you.
I've been here too long. Here.
Mhm.